Thank you everyone for joining today. My name is Ross McFadgen. I'm the product manager at Diagnostic for our molecular portfolio. And today I'm going to present you on a basic introduction to NGS data analysis. So this is a three-part series. My colleagues have already presented on the start of the NGS workflow from extractions to QC, right through to library preparation as well as sequencing. Today's webinar is going to focus on really everything that happens after the sequencing has performed. So once your data is ready for analysis, and we'll basically cover these three different sections of the analysis workflow. And we'll also touch on two of the platforms that we supply from Diagnostic, which enable NGS data analysis. So if we take a very high look level at a NGS workflow for data analysis and what comes after the sequencer has actually run. So we have our sequencer that has produced a signal to read the nucleotides of the libraries. This signal needs to be converted into FASTQ files, which are file types that can be read by downstream software. The data coming off the sequencer is raw data and can't be interpreted without converting into FASTQ files. Once we have FASTQ files, those need to be aligned to a reference genome, which will allow us to identify any differences between the reads of our samples and the reference that we are using. Finally, then, after the alignment, we start to call the variants and actually start to interpret what effect these variants will have in terms of their clinical significance or just functional effect on the proteins. So when we can break this workflow down into several different steps, and I'm going to go into a lot more detail on all of these a bit later, but the easy way to see it is essentially the primary analysis, which is handled by the sequencer, the secondary analysis, which refers to the alignment and variant calling, and then the tertiary analysis, also called clinical interpretation. And this is really finding out what effect that variant has on your sample or your patient. So to start off, I just want to cover some of the basic file types and nomenclature that we deal with when we're looking at NGS data. So I'm sure many of you are aware of FASTA and FASTQ files, but just to really show you what the difference is between those. So a FASTA file is something you would get off doing a Sanger sequencing workflow, or many genes are also coded into FASTA files. Generally, you'll have a header, which will give the organism or the gene name, and then just a continuous stretch of nucleotides, which really show what nucleotides are contained within that gene. On the right, we're looking at a FASTQ file. The major difference here is that the FASTQ file contains the sequence reads as well as a corresponding quality score. So if we look here, this is the actual read, and then under each nucleotide, we have a symbol. And this symbol translates to a Phred score, so a Q score. And it's coding using ASCII characters. So just for example, an A in this case translates to a Phred or Q score of 32. And FASTQ files are usually zipped since they're quite large file sizes, often using a program like gzip. And you can just use a simple Windows program like WinRAR to extract this into the main file type. Taking a bit of a deeper look, here we are looking at a real FASTQ file that was generated, and we can see it's quite a large size, so around 3 gigabases for this one FASTQ file. Now, you can actually open FASTQ files in a text editor, so something like Notepad++, and this will allow you to actually see the contents of the FASTQ file. So for example, if we take this file and we open it up, this is what we see. And there will be thousands to millions of lines within each FASTQ file, depending on how many reads were generated. But just looking at one of the lines over here, and let's go over what we're actually looking at. So firstly, each FASTQ file will have a header, or each read within the FASTQ file will have a header. And on the right here, I've kind of just shown what each of these things mean. And our different sequences will have slightly different headers. So for example, in this case, the first value is the name of the sequencer. Then we have the number of runs that sequencer has performed, the ID of the flow cell, which lane it was run on, and then the actual coordinates, because remember, each library is sequenced from a cluster or colony on the flow cell. And for us to call those bases, we actually need to know where it exists on the flow cell. And then we have the number of the read, and then finally the index or barcode. And now you can see in this case, since it's the same FASTQ file coming from the same sample, they contain the same index. So the indexing is really important in NGS because that allows us to tie specific reads to a specific sample. And then just looking at the contents here, again, we have the read length. So if we count all of these characters, it's probably going to be 150 base pairs because this was a 150 base pair sequencing run. And then we have the Phred quality scores for each of the bases that were called in that read. So moving down the analysis workflow, one of the next file types you would deal with are either SAM or BAM files. Now, a SAM file means a sequence alignment map. And this is really a text-based file that is used to store sequence data, as well as mapping position and quality scores. So it really deals with the alignment of the reads to the reference, as well as the quality of the mapping at those positions. Now, a SAM file is something we call a human readable file. This means you can actually open up the file and you can read it. The other file type called a BAM file is a binary version of a SAM file. So it is a smaller file, it's compressed, and it's non-human readable. So you can't just open it up and view it on your computer. This is typically used as input into a variant calling pipeline because the BAM file is collection of the reads aligned to a reference. And now with variant calling, we want to be able to call variants which are different to the reference genome. And if you have a BAM file, you can often open these up in genome browsers such as IGV, and that's what we're looking at on the top here. This is taking a BAM file, opening it in IGV, and now we can actually see each of the reads that were generated and where they are aligned against the reference. Another file type that you'll deal with in NGS is called a bed file, which is a browser extendable file. It's tab delimited, and essentially it's quite a basic file. At its most basic, it only needs 3 fields. That would be the chromosome, a start and a stop position. And what it really is just a way to kind of define targets, either within an NGS panel or within an analysis workflow. So for example, where we use these is if you design a custom NGS panel, or even using an off-the-shelf panel such as an exome, we want to know the specific targets within that design. So we use the bed file along with the BAM file to direct the analysis to a specific section of the genome. Because you can think of it in this way, if you do an exome and you do an alignment, you know that your data only represents 1% of the genome. So you don't want to align an exome to a genome because it's just going to waste a lot of computational resources. So using a bed file, we can tell the analysis only focus on these specific portions of the genome because that is what the panel that I use to do the sequencing contained. And then finally, the variant calling file, the VCF. This is really generally the end of a variant calling workflow. A variant file will be generated. And what it contains, obviously a really big header. The header is just really metadata, information on the filtering and stuff like that. But the real core of the VCF is contained within the body. And I'm going to show you another one in more detail. But really what we're seeing here is a specific position in the chromosome, a SNP ID if it exists, the reference and the alt allele, and then quality metrics and filters. So it's really telling you all of the variants that were identified in that sample, along with additional metadata that can be used downstream in the analysis. So just some other nomenclature I want to cover quickly on target rate. So this is really if you're doing target capture. So again, exome panels from say Twist or Agilent. The on-target rate is telling us what percentage of the total reads map to our region of interest. And typically it's expressed as a percentage. And what it's calculating is how many of the reads are actually outside of that region. So for an exome example, we know the probe should be on all protein coding exons. But typically we do get probes binding to off-site targets, and that isn't calculated into the on-target rate. Another thing to mention is coverage uniformity. And what this means, it's really describing the read distribution along those target regions. So if you're looking over here, these black boxes are, say, genes or exons that we are trying to target with our panel. And what we are seeing here is that some genes get sequenced above the depth that we require, and some get sequenced under. And this is really what we talk about when we talk about uniformity. In an ideal world, 100% uniformity would look like something at the bottom, where every target is sequenced to a perfect depth. However, this is not true in the real world. We always see a bit of non-uniformity. Some genes need to be sequenced a little bit more and some a little bit less. but just something to be aware of when analyzing a target enrichment panel and what to look at. And just to go on from that, just to show you some examples of depth versus uniformity. So at the top one, we have good sequencing depth and good uniformity. So that depth is maintained across the entire target region. And that's really an ideal world. The one in the middle is low depth, good uniformity. So we can see it's uniform, although there's less reads than we would like. So the read depth is a bit lower. This is an easy fix. We know the panel is working well because we have high uniformity. All we need to do is sequence it a little bit deeper. So either add less samples onto the flow cell or use a bigger sequencing kit just to up the depth. Typically what we see is, typically what we see is not poor uniformity, but a mix of good and bad. So where we have some regions very highly covered and some almost not covered or very low. So it's all about really optimizing the uniformity of your target enrichment panels. And then finally, Q-score or Phred score. If you deal with NGS a lot, you'll be quite familiar with Q30. Q30 is like the gold standard for NGS data quality and typically represented like this. So it would say more than 90% higher than Q30. So that's telling us that of the data that was generated, more than 90% of it has a Q-score of 30 or higher. And when we talk about Q-score, it's really a confidence score. How confident are we that the base at that position is called correctly? And it's not just used for base calling, it's also used for mapping and any kind of quality predictions. So how we can see a Q-score is read here. I'm just going to start with a Q of 30. This means we have a probability of one incorrect base call in 1,000 calls. So that's an accuracy of 99.9%, which is still extremely high. But today, and with the advancement of NGS technologies, we're actually seeing more Q50 and even Q50 data with some special types of runs. So when we refer to Q50, we're talking about one error in 100,000 base calls. So a 99.999% accuracy. And then just another differentiation I want to make between sequencing and what we call a counting application. So in traditional variant calling pipelines, we use reads of about 100 to 150 base pairs, and these are then aligned to a reference genome, and the goal here is to determine genetic differences. So we want to be able to call variants from those reads. But when you're doing RNA-seq for gene expression, the goal is slightly different. Now we want to be able to count how many transcripts are expressed for each of those genes. We don't really need to call variants in those reads. So this means that we can use shorter reads. Typically, RNA-seq reads are around 50 to maybe 75 base pairs, because we only need enough read to map to the reference so that we can call what gene was sequenced. We don't need very long reads because we really just need to be able to count. And this also applies to, say, single-cell RNA sequencing, for example, 10x Genomics. They use a 90 base pair read for their transcript read, because again, that's long enough to map to a reference to get an accurate call of what gene it is. And therefore we can count those transcripts, because the end goal here is to calculate differential gene expression and not to map and call variants. And typically this is expressed as a volcano plot, which you can see at the bottom here, showing upregulated and downregulated genes between two different samples. So now I'm going to take a bit of a deeper look into this whole workflow, starting from the primary analyses. So as I mentioned, the goal of the primary analysis is to take the raw signal from the instrument and then to convert it into base calls. And different platforms do this differently. And I'm just highlighting these two well-known examples here. So Ion Torrent, they use a pH change to actually calculate what base is called at that position. Whereas platforms like Illumina and Element and MGI, they use fluorescent imaging. So an image is taken and a base is called, but the end goal is really the same, to call a nucleotide base. Down the primary analysis workflow, we generate FASTQ files, we do some additional filtering, the demultiplexing, and then trim adapters. And this gives us the FASTQ file. FASTQ file is then inputted into a secondary analysis pipeline. And this is really where we do the alignment to get the BAM file. We then can do some post-alignment processing, call variants, and generate a VCF file. VCF file is then used as input into our tertiary analysis pipeline. And this is where we try and figure out what impacts the variants have on the sample or the patient, whether they are real, whether they are pathogenic or not. So we go through steps of annotating the variant to determine its functional effects. We then filter those variants based on quality, population filters, and things like that. And then we actually interpret. We take the candidate variants and really try to figure out which one of those variants best matches the patient phenotype. And typically, this is then a clinical report which is given. So if we just consider the output being a FASTQ file, a VCF file, and then finally a clinical report. So I'm just going to look at each of these in slightly more detail. So for primary analysis, as I mentioned, this is taking raw signal data on the instrument and converting it into sequence data, showing these two different examples. This typically happens on board the instrument and is not something that the user needs to really tweak or be a part of. It is typically a default process that happens based on the parameters of the sequencing run and usually includes some amount of pre-processing. So this is just filtering out really bad quality reads, maybe reads that are too short and don't meet the read length. The demultiplexing is important. Obviously, with NGS, we multiplex a lot of samples onto a run, and now we need to demultiplex and assign reads to each of their unique samples. Then finally, we do trimming, and this is just to remove adapter sequences since we don't want those to interfere with the alignment. And we can also filter for bad quality sections of a read to remove those before you do your alignment step. So it's just a way to really trim the data to get it into a better format before we do alignment. And typically after the primary analysis and demultiplexing, you can take your FASTQ files and use a program like FastQC, which is a really nice, simple program that allows you to input a FASTQ file and then go through each of these different parameters. So for example, here we can see the per base sequencing quality started off with a Phred score quite high and then dipped quite low. And that's why it's giving an indication of a warning there. So FastQC is a nice way to just look at the quality of your FASTQ files. It doesn't really say anything about the sequence content or variants. It really is just about the reads themselves. And now we're going to move over onto alignment. So if we think of the aim or the goal of sequence alignment, we've now got these reads that have been generated and converted into FASTQ files, but the reads themselves are just collections of nucleotides. They don't tell us anything about which gene they're associated with. So an alignment program aligns those reads to a reference so that we can tell, firstly, which gene it is from, and then secondly, identify differences between those genes. But there's quite a trade-off, and there's many different alignment tools, and it's really between speed and accuracy. And this is going to really depend on what type of application that you're doing. If you think of old school traditional methods like BLAST, there's no way a human or even computer programs could sit there and do an alignment through BLAST. If we think of a single human exome containing 48 million reads, and this is why we need these alignment or mapping programs. And we can really break it down into two separate methods. So something called resequencing, which is where we map reads to a known reference. And this would be obviously for humans using HG19 or HG38. We do a run and we map to the human reference since we know our samples are from a human. Obviously, if it's from a mouse or another organism with a reference, we would use that corresponding reference. There's also a de novo assembly. This is where we don't use any reference. So we get the reads, but we don't align them to a reference. We compile those reads into a contig. This is typically done for organisms that might not have been sequenced, so there's no reference before, or you just want to build a genome. And this is just an example of some of the mappers that have been out throughout the years. So we're looking at DNA mappers, RNA mappers, microRNA, and bisulfate mappers. So again, you need to select the right mapping algorithm for the type of data that you're dealing with. Some of the common short read mappers would be BWA-MEM. This is for RNA or DNA-seq, 75 base pair reads. Bowtie2, which is a really fast and memory efficient mapper. TopHat2 is specifically for RNA, and this is used to identify splice junctions between the exons. And then something like STAR, which is for big bulk RNA-seq analyses. So the choice of mapper is really dependent on your sample type and the question at hand. And what we're looking at here is really reads aligned to a reference genome. So just breaking it down, at the top, we have the chromosome, and we've highlighted the specific band. And within this band, we have these two genes. So we can see the reads are color-coded, representing the forward reads in red and the reverse reads in blue. And this shows how we can tile over a specific section of the genome. And what we want to point out here is a specific variant that is actually being called. So if we go down here, we can see these lines in the reads, and these indicate to us that the base call at this position is different to what the reference genome calls, which therefore means we have a variant. And if we zoom in to this specific variant, what we can see here is we have an A to G variant at position 923 in the cDNA. And this is quite clear. If we start to look at the reads here, we see that the reference should be an A, but we are seeing a lot of Gs being called both in forward and reverse reads, which means this is a true variant. And we can verify that by looking at the variant allele frequency. So we've got A at 56% and the G's being called at 44%. So this is a typical heterozygous variant, almost a 50-50 split between the two alleles. And I just wanted to include this to really show the difference between mapping with short and long reads. So the most of my talk is really focused on short read sequencing, but this is really just a nice visual cue to show you why long read is sometimes preferred over short read. And again, we're looking at a specific gene at the top here. The exons are these bars over there, and the introns are the line. What we see with short reads, obviously comprised of many, many short fragments, it's able to tell quite well over the gene. We do see some regions with higher coverage and some with lower coverage. But the main feature here is that we do have unmapped regions. This can be due to difficult to sequence regions. Maybe they have a lot of repeats or other issues which short read sequences can struggle with. If we compare that to the one below, which is a long read platform such as ONT or PacBio, we now see that the reads themselves almost cover the entire length of the gene. And this is beneficial for specific applications, one example being like HLA typing, where we want to know the phasing referring to SNPs that fall on the same chromosome. With short reads, it's hard to tell that. It can be done computationally, but it's still a bit of guesswork because how do we know that those two reads are from the same chromosome? When we have one long read covering the whole gene, we know that is from the same chromosome so that these SNPs can actually be phased and tell us that they occur on the same chromosome. So when you're doing inheritance patterns, we can now know the phasing of those alleles. So moving down the workflow, we're now looking at post-alignment processing. So once reads have been aligned, there is still some processing that happens. Firstly, duplicate removal. So sequencing duplicates are generally from a PCR. And what it means is we have two reads that have the exact same start and stop coordinates. Typically, this is caused during overamplification step or something like PCR bias, where we have one fragment that keeps being copied. But we need to remove these duplicates. The implication, if we don't, is that you'll actually artificially inflate the presence of a mutation because you're essentially artificially inflating the copies of that genome since those reads are just duplicates of each other. And this can lead to false positives or skewed allele ratios. Typically, we want duplication rates to be less than 10%, and that's kind of the goal that we aim for. Once duplicates are removed, we do some local read alignment. This is just an algorithm that improves the accuracy specifically for indel detection and reduces mismatches. And then finally, a final base quality score recalibration. So again, I said earlier that Phred scores are also used in mapping. So it basically recalibrates those scores to make sure that you're getting the best quality match for that read at that position. All right, so once we have aligned, we are now ready to actually start calling variants within the genome. And the aim here is to identify polymorphic sites, so nucleotides that differ from our reference sequence. And looking at this example here, we can see that the reference at the bottom at this position should be an A, but we have mostly G's being called. So this could be a homozygous variant where every position called is different to the reference. But what we also typically see are these ones that float around randomly. And these are generally sequencing or PCR errors. And we know that because they only appear once or twice in the reads. If we see that it's occurring, at quite a low allele frequency, it could be a somatic mutation. But generally, if it's popping up once or twice, this is just an error and they can be ignored. But the goal here is to determine the different nucleotide, but then also use statistical methods to tell us, is it actually a true variant? Or is it something like just an anomaly in the sequencing? So when we look at variant calling tools, there's many out there, many well-known ones and very specific ones as well. Often you need to know what you're looking for. Some pipelines are better than others at doing specific functions. So in terms of what we're looking for with variants, is it SNPs or small indels, less than 50 base pairs? Are we dealing with maybe large indels greater than 50, structural variants such as CNVs or other complex translocations? On the right here, I've just shown some examples of tools that are used commonly, as well as what platform they run on. And that's another important point to consider. If you want to run Samtools, you obviously need to run it on a Linux platform. Some of them have compatibility with multiple platforms, but just something to be aware of. GATK is a very widely used tool. It's very scalable, and it's focused on variant calling and genotyping. So you might have seen this commonly used even in a lot of platforms that offer variant calling as a service. In the background, they're probably running some version of GATK. And just to differentiate between two different files, so a raw VCF would be pretty much what comes out with no filtering. So it would list every single variant detected from the reference, many of those being benign, many of them being low quality variants. The filtered VCF is where we use pre-filtering for quality, read depth, allele frequency to really contain only the the variants in the VCF that we think are actually true variants. So now looking at a VCF file in a bit more detail, I'm just highlighting this one line at the top. So these are all from the same sample, but we're looking at different variants at different positions in the chromosome. So the first field is telling us this variant is on chromosome 20 at position 14,370. This field here is pulling through the RS SNP ID. You won't always see this because sometimes there isn't a SNP ID or an RS number for a SNP. But if there is, the variant calling tool can actually pull that through and tell us, okay, this SNP is actually a known SNP, and here's its RS ID. We then obviously get the reference sequence and then the alt sequence. So this would be the alternative allele. When we get the Phred quality again, so this is a quality of 29, which is really good prediction. And the filter here is telling us that this passes based on the quality and allele frequency. And then we have a lot of other metadata here being the depth, the allele frequency, and so on. Just for example, you can see this variant, although it was called very low quality, and it's actually failed the filter. And the reason for failure is that the quality is less than a Q-score of 10. So this is probably not a true variant. So now that we have aligned our reads, we've called variants, we've identified which variants exist within the sample, the real tougher part now is to determine what effect these variants are going to have on the transcripts, on the proteins, and whether they are actually pathogenic and match the phenotype of your patient. So we can think of this as a type of triage, right? So we're starting with thousands or tens of thousands of variants, and we need to get down to a handful of candidate variants that we think are the actual variants causing the effect that we are seeing. There's lots of tools that can be used for this. There are online platforms, which I'm going to show you in a bit later, that can do this for you. But the idea is to really compare the variants to multiple databases and use in silico prediction to determine the functional effect and really how rare this variant is in the population. So ideally, if you're doing something like an exome, you're going to have 50 to 100,000 variants. It's impossible for a human to work through each of those and basically start to cancel out or not. And this, you'll see later why we use these online tools to do this for us automated. So although there's these platforms I'm going to show you in a bit, a lot of them are running these types of tools on the back end of the platform. So once we have our variant, there's many ways to figure out how pathogenic it is. So firstly, we want to determine the functional effect. And here are some common tools that are used that are basically in silico tools that are predicting how bad the functional effect is on the protein based on the variant that has been called. So whether it's a missense variant or a nonsense variant or loss of function, it's going to tell us what prediction of pathogenicity is there due to that variant. We then also use a lot of databases, obviously ClinVar, which has lots of variants that are known to cause disease, dbSNP, which has a range of known SNPs, dbVar for structural variants, and then things like OMIM, which have a curation of variants and human genes and phenotypes. So these really tell us, have these variants been identified before? Have they been associated to diseases? And this can help us kind of predict and come to an answer. Other very common and helpful databases are population allele frequency databases, such as 1000 Genome and gnomAD. And the idea behind this is that if we detect a variant in a population, but over 50% of the world has that same variant, it's very unlikely it's a pathogenic variant since half the world has it. However, if we detect a pathogenic variant that is less than 0.1%, or maybe not even in these databases, that tells us we've detected a very rare variant that has not actually been seen before. And then you would start to look at it in a bit more detail. And then there are more specific databases for oncology, such as COSMIC, and pharmacogenomics, such as PharmGKB. And these will really have a bit more detail on variants that cause known cancers and treatments and things like that. So what I want to move into next is really showing you some of the platforms that we distribute for in South Africa, starting off with SeqOne. So SeqOne is a cloud-based platform, and it really does secondary and tertiary analysis. It's a nice online, user-friendly, very comprehensive platform. And I'm just going to run through one example for you today, just to show you some of the features, how it works, and how we can call pathogenic variants based on the databases. So just some quick background. SeqOne is based in France. They're a cloud-based bioinformatics platform, and they offer comprehensive analysis for somatic, germline, and infectious disease. They've grown quite a bit in the last couple of years, and if you hadn't heard recently, they acquired both Congenica, which is a very well-known data analysis company, as well as Life and Soft, which are infectious disease analysis company. So those were both acquired in 2025, which is expanding the features of their platform. They support both secondary and tertiary analysis of raw sequencing data and CGH microarray data. So again, this means we can start from a FASTQ file to do variant calling and the clinical interpretation, or if you already have a VCF file and you just want to nail down a pathogenic variant, then you can only run the tertiary analysis. So it's a very comprehensive but flexible platform. Provides you with an interface to view and interpret the results with the end goal of finally generating an official clinical report. It uses the ACMG rankings and guidelines, as well as a wide range of integrated annotation bases, some of them that I've just presented to you. And the best thing about it, it is platform agnostic. So whether you're working with iron torrent data, MGI data, Element, Illumina, SeqOne is able to handle all of that data. And it supports analysis from gene panels, exomes, whole genome, and RNA-seq. Another great feature of SeqOne is that there's no annual license fee. You pay for what you need. So you purchase credits to use for your specific run, and there's no need to pay a license fee every year, even if you aren't really using the platform. So when we look at their current solutions, they really have quite a full spectrum from oncology to rare disease. So starting on the left here, these would be some of their somatic pipelines. And these are obviously used in oncology to identify mutations in tumors. So we have the solid tumor pipelines, liquid biopsy pipelines, the hematological pipelines, and then hereditary cancers. For their germline applications for inherited disease, we have some rare disease, inherited disorders, carrier screening, as well as hereditary cancers. And then finally, the newest addition to SeqOne being an infectious disease portfolio. And this is for detecting microbial pathogens and doing things like drug resistance calling. So it can handle viruses, bacteria, AMR, as well as gene function prediction. For today, I'm just going to focus on one of the germline applications. So this is some real data generated by a customer of ours. They were running the Agilent V8 exome, which is the latest exome panel from Agilent. And this was sequenced on our Element VT platform. And just to quickly note some of the variants that SeqOne can deal with. So SNPs, indels, larger indels, CNVs, structural variants, ALU elements, multinucleotide variants, RNA SNPs, gene fusions, and then biomarkers such as MSI, TMB, and HRD. So what we're looking at here is really the first step of variant interpretation. This is after the tertiary analysis has completed. And I've just taken a snapshot of 8 of the top variants. So this is one sample and all the variants. And this is just it continues on the page. So this is the same 8 variants, just the end of the page. I couldn't fit it on one screen. So let's go through some of these things at the top. Firstly, we've got the transcript. So obviously some genes have multiple transcripts, and this is where you can actually choose which transcript. For example, this is telling us there's 3 different transcripts that could be used here, but we are showed here the transcript that's being used for the analysis. Here we have the DIAG AI score, and this is a sequence-specific predictor. I've got some slides in a bit, but it's really using AI to do a prediction on the pathogenicity of this variant and whether it applies to the phenotype of the patient. We obviously have the ACMG rankings, and then something called the variant knowledge base. This is for your own account, so it's your own variant knowledge base. As you can see, these are all new. They've never been detected before. But what you can do is actually add a variant to your knowledge base. And this obviously helps in certain scenarios, especially I think in South Africa, where, as you know, a lot of our work is aligned to a European reference. So some variants might not make as much sense when we're using, say, African-derived samples on a European reference. So you can start to actually collate variants that you detect into your own knowledge base so that you can see when they appear again. We obviously have the gene that this variant is detected in, the transcript. We have the nomenclature of this, this variant. We can see here it is a deletion. It causes a frame shift, which is a high functional effect, and it exists with an exon 4 out of 17 in this gene. We can then tell the transmission of this as autosomal dominant. We can link it to the phenotypical HPO terms. And then finally, we can see where this has appeared on Panel App, which is a curation of panels and variants from Australia and England, showing variants that are known to cause disease. Continuing on that window, we get then the ClinVar reference. You'll see in this case, there wasn't actually a ClinVar reference for this top gene. It also did not exist on the gnomAD population frequency database, meaning it's a very rare variant. If I just go one down, you can see this variant was in ClinVar, 2 variant of unknown significance, and it is contained within the gnomAD database. We then have the sample frequency, and this is just within this project, how many times this variant has been detected. The prediction scores, so here we can see pathogenic. I will speak on this in a bit more. And then finally, just the sample information. So variant allele frequency and the coverage, as well as the genotype, so this being heterozygous. So what I'm looking at now is one specific variant. We have zoomed in on this top variant that was called this frame shift deletion in DDX41. Firstly, I'm going to start with the ACMG rankings, which most people would be familiar with. So you can see here, there's a tab that you can move along for each variant. So I'm in the ACMG tab. This has been predicted as pathogenic using automatic tagging, and we can see why it's been called pathogenic. So we have this field, PBS1. It's predicted a null variant in a gene where loss of function is a known mechanism of disease. So obviously a frame shift mutation is going to cause a loss of function, which means this protein is not going to work, and this protein has been associated with disease. So that's a very strong evidence for a pathogenic variant. The second one, which I touched on earlier, is that this variant is absent in population databases, which means it has very likely not been seen before, meaning it's a very rare variant with a functional effect. So again, another big association to pathogenicity. And then we're moving on to the DIAG AI. So this is really SeqOne's AI prediction tool. And that's where these 3 windows here come from. So starting at the bottom, when I click DIAG AI, it's going to open up and give me the DIAG AI score. This is a score from 0 to 100, 100 being the most pathogenic. And it's using certain in silico tools and databases to basically call whether this gene is pathogenic or not. And we can tell how it is scoring those, as you can see here. So we've got a pathogenicity score using the universal pathogenicity predictor of 33 points. And we can actually calculate the impact based on these scores. So as you see here, the predicted functional impact is very high. That's the largest contributor to the pathogenicity score. We then have the population frequency. It's absent in the database. Again, that means it contributes to the pathogenic score since it's a very rare variant. And then going down, we can see different fields that it's used to assess the pathogenicity, such as the number on the exon. Obviously, if it starts early on in the exon, that gene is going to be complete loss of function. Maybe if it was at exon 16 or 17 towards the end of the gene, it wouldn't be as bad. And then a range of other features there. So it's really not a black box. They're telling you exactly how they predicted the pathogenicity. And then in addition to that, we also have these score boosts. So it uses certain things to boost the pathogenic score based on stuff like transmission mode, quality checks of the variant, looking at sequencing depth, allele frequency, whether it fits the inheritance type of this variant, as well as panel app. So combining all of these things together, it's able to give us this DIAG-A score of 80, which was the highest score for this patient, the highest variant score, meaning that this is very likely the variant contributing to the phenotype, and that also ties in nicely with the ACMG rankings. Just quick information on DIAG-A. I have just pulled this from the white paper, so if you do want to know any more information, SeqOne have a really nice white paper, but essentially it's what I've explained to you, a score from 0 to 100 that indicates the likelihood of a variant being disease-causing. And the score is integrating the pathogenicity assessment, phenotypic relevance, and then those expert rules and boosts based on inheritance, quality, and gene relevance. And really at the bottom here, it shows you, so for pathogenicity, it's been trained on over 2.5 million ClinVar variants using 72 different features to predict the pathogenicity of a variant. Then something called PhenoGenius, which is using the phenotype to be able to match phenotype to a variant using more than 6.3 million interactions between 18,000 phenotypes and 6,000 genes. And then finally, the rule-based system, which is just using those rules to really look at quality and inheritance and to add to the score of being a real true pathogenic variant. So now moving on, we're looking at the CNV tab. This is actually for that same patient. So on the left, we're just seeing the main CNV window. Here we can see it's showing us 15 out of 259 CNVs that were detected. I can then use some filters. In this case, we're saying only show them the OMIM morbid genes or CNVs. We can then filter on size and sample frequency. I haven't used any filtering here, so we're just looking at the OMIM morbid genes. You can also do your own gene panel or just search for specific genes if you're trying to target your analyses. So if we look at this specific CNV, it is quite large, about 7.4 megabases at chromosome 6 on the Q arm, and it's got an ACMG ranking of 5. So again, very pathogenic. What type of CNV is it? It's a 1 copy loss indicated by the down arrow and the -1. And we see here it has a really high quality score. So this means it is definitely a true CNV, and it contains 4 OMIM genes, so 4 clinically relevant genes within the CNV. If I now open up this specific one, we get a bigger summary of the CNV. So firstly, what it's telling us is this CNV exists in a clinical region. There are 4 out of 36 clinical genes that are covered by this CNV, and it overlaps with this database at 1.6%. Now we can look at the pathogenic call. So why did it call it as pathogenic based on CMG guidelines? And here we can see the scores that it gives based on the copy number, as well as the genes or genomic regions that's associated with. It then shows us the clinical region. So here it's actually given a name for this deletion, showing a 33% overlap with that OMIM record. And then it's looking at the clinical genes within that region, as well as the phenotypes that have been seen for these CNVs. Another really nice feature is at the top here. It can link out to UCSC, Decipher, as well as IGV. And SeqOne actually has IGV built in within the platform. So if I click on IGV, it's going to open up a window in SeqOne without having to go to another application. And now what we're seeing is what you would, if you do CGH type of analysis, more of a common type of view where we're looking at chromosome 6, this specific portion of chromosome 6, and then we're looking at this little band over there. It is, as I said, quite a large region of about 7 megabases. And what we are looking at here is the diploid copy number. So obviously being humans, we have 2 chromosomes, so we should have 2 copies of each gene. And that's indicated by these green dots. And these are the probes that were actually used, and we can see the genes at the bottom. So this is normal, being 2 copies. And then we see this drop off here for this section, where we only are seeing 1 copy of the gene throughout all these different genes, right until here where it goes back. So this would be quite a big 1 copy loss of this large portion of the genome. And a great feature of SeqOne is their built-in QC. So once your run is uploaded and analyzed, the first thing you want to do is obviously look at the quality to determine if there's any dropouts, or maybe you need to sequence more, or the quality wasn't good enough. I'm just going to walk you through some of the basic QCs. So here we're looking at this one sample. It's from an exome, and here are the different QC metrics. So things like, is it the correct sex based on what was provided and what chromosomes were detected? Is there any contamination from other genomes? What is the percentage on target, coverage depth, percentage of duplicates, and so on? And I've just highlighted a couple here. So if you click on any one of these, it'll open up another window. And I just want to walk you through a bit here. So remember, I was referring to percent on target earlier in the talk and how that basically tells us what percentage of the reads are aligning to the target of this panel. Typically, you want this above 80%. So you can see here we got a warning, and that's telling us it's between 80 and 84%. Anything less than 80 would be a fail, and higher than 84 would be passed. So warning is not a bad thing. The analysis will still go ahead. It's just letting you know, hey, your on target is slightly lower. I wouldn't be concerned here since we had 83.1 and the cutoff is 84. We then have percentage duplicates. So remember, I said we generally want, 10% is good. Here you can see the warning is between 20 and 40, and a failure is anything more than 40%. And just to highlight here how low our duplicates actually are, and this is primarily due to the VT sequencing chemistry. So we see much lower optical duplicate sequencing on a VT, and this is showing you right here, only 5% of the reads were called as duplicates, which is quite a high pass if we consider that 20% is the start of the warning. So very, very low duplicates, and that means you're not filtering out a lot of your data. We're then looking at coverage depth. So the pipeline wants a depth between 70 and 30 as a warning, above 70 as a pass. Anything less than 30x would be considered a fail. Obviously, not enough read depth to accurately call variance. In our case, we got 151 mean coverage, which is almost double the threshold of the platform. So we have plenty of data to accurately call variance. And then the final one is just the Q30 on the reads, so percentage base pair Q30. Again, we got a warning, but it's just because SeqOne used extremely stringent thresholds. So 94.8% Q30 is a really good high quality run. We're getting a warning because the cutoff is 95. So we're just on the border of that cutoff, but anything higher than 90% would be a warning and anything lower would be a fail. So it's still really, really good quality data, even though we got those two warnings. Going further down the QC tab, there's some more general QC. So things like coverage, GC content, and Q30 scores. So the top bar is telling us percent coverage at different depths. So how you interpret this, at 50x, 97.66% of the target was covered. At 100x, 82% was covered, and so on and so on. So this is really good. Generally, you want 99% at 30x, and that you can see we have here. We're looking at the GC content. So we have what it theoretically should be, as well as the GTC content of the reads. And then finally, a typical Q-score graph showing us the read as well as the Q-score. We can see this was sequenced on a VT because our Q-score is sitting at about 40 throughout the entire read. So again, very high quality data and no drop off at the end of the read. On the right-hand side is just some basic alignment metrics that we get. So the total number of reads for this sample, median insert size, read length, how many aligned, base quality, and average coverage. But a really cool feature is this gene exon coverage window. So this would list every single target in your panel. So for an exome, that's over 18,000 genes. And it's showing you what's been covered and where we have warnings. And that's based on those same coverage values that I showed you earlier. So this just means for this portion of the exon 1, it's covered less than 70. Again, it's a warning. You don't have to worry. You can go ahead. But as we see for most of the genes, we have full coverage in the targets, indicating that we have high quality, high coverage data. And now I'm just showing you some other features. This is not related to the exome that I was showing you. This is actually doing an oncology panel. I just want to show you what else SeqOne can do and some of the visualization tools that it has. So obviously doing fusions, this depends on the panel you're using and whether it can detect fusions. But if you're using something like Agilent's CGP panel, which has a 80 RNA gene fusion module, we can detect novel fusion genes without even knowing the partner. And what's really nice about SeqOne is it gives you this nice visual representation of the breakpoints between the genes, and then actually shows you the two different genes and how that would look. In terms of biomarkers, again, you would need to use a panel that is of the right size to call biomarkers such as TMB, so generally bigger than 1 megabase, you can call tumor mutational burden. And here it's showing us the coding region size was 1.7, so that's more than enough to call TMB. And here we got a TMB score of 24.6 mutations per megabase. And we also did the MSI, microsatellite instability, and here we can have an MSI, it's a moderate score of about 30%, giving us the MSI score and the MSI status. And then just some other general features. This would exist through exome or oncology analyses, but things like filters and ranking. So it's nice that you have these automated rankings such as DiGA-I and ACMG, but you might want to dig a bit deeper and play around with your data. So there's a very comprehensive filtering tool on the platform, looking at things like specific genes. You can filter on the quality and the coverage, allele frequency, things like sample frequency, the impact, the effects. So you can really, dig deep and kind of set your own parameters to look for a specific variant of interest. A very nice feature is these in silico panels. So for example, you sequence a whole genome or a whole exome, but you're actually only interested in about 50 genes. So instead of running the analysis on all 18,000 genes when you only want 50, you can create your own in silico panel. As you can see here, if you click on the drop down, these are some that have already been created. You create a panel, and then that will be available for you every time you run the analysis. You won't have to type in your genes each time. So this is a nice, quick, and easy way to just focus the analysis on a subset of genes of interest. We then can also see the databases that we used and the different calls within those databases. This obviously exists for the sample as a whole. So in this case, the sample had 71,000 SNPs that were represented in dbSNP, 35,000 variants that were represented in COSMIC, and so on. And then finally, the rankings. So the ACMG rankings here, in this case, 4 pathogenic variants were identified in this sample, but you can see the vast majority of variants were identified as being benign. Almost 62,000 variants in that patient were benign. So that was SeqOne. I want to just briefly move on to another analysis solution that we support called HyraxBiosciences. They are actually a local company headquartered in Cape Town, and they developed a tool called Exatype. It's also a cloud-based DNA sequence analysis platform, but it is really designed for infectious disease and specifically drug resistance profiling. So what's nice about Hyrax, as I said, it's an online tool. You don't need any training. It is very easy to use and can run multiple different types of analyses. Because it's cloud-based, it's very scalable, meaning you can throw a lot of samples at it without having to worry about analysis times, since no hardware or software is needed on your side. This can actually integrate directly into a sequencing instrument or LIMS system. And this means once your sequencing is finished, it automatically is going to be streamed into Exatype for analysis, and the user won't have to copy or transfer any data. It's very extensible. So although they have a set of developed pipelines, which I'll show you in a bit, they can actually create tests for anything. As long as there is a reference genome, it obviously comes at a fee, but they can set up, for example, if you want a monkeypox analysis pipeline. They don't currently have that, but they can set that up and just develop that within Exatype. So these are the currently available analysis solutions. So Hyrax and Exatype started off with HIV analysis. That's what they were really known for. So they have an NGS-based HIV genotyping solution, as well as a Sanger-based HIV genotyping solution. During COVID, they added on the SARS-CoV-2 NGS analysis. So this is for variant typing and lineage analyses. And then 2 other solutions would be the HIV NGS proviral solution, as well as their MTB NGS solution. The MTB solution works for both targeted and whole genome. So this means if you're using, say, a targeted panel only looking at drug resistance genes, we can analyze that. Or you've sequenced the whole genome of MTB and you want to be able to call antibiotic resistance and do some lineage typing. Just showing you some of the outputs and reporting on Exatype. So obviously nice actionable report showing drug resistance and the drug class, as well as the variant that was called. Lots of cool customizable outputs. So these are like number of reads per sample, showing forward, reverse, low quality, not aligned. This one here is showing the read coverage over the SARS-CoV-2 genome. So here's the entire genome length. And here we're looking at the min and max coverage, so the number of reads along the genome. We see some regions much more covered than others. At the bottom is the drug resistance report for MTB, which I'll show you in another screen. A nice feature is that they allow you to download all the raw data. So it's not just that you put in data and get out a report. You can download the consensus sequences, the aligned BAM files, the coverage reports, the raw data. It's all available to you within the platform. And then, as I mentioned, this can all integrate with LIMS or databases that the user has. So this is just looking at an MCB drug susceptibility report. We have some sample names here, what pipeline was used, what it was sequenced on, what organism it is, as well as the drug resistance mutation prevalence cutoff. So this can be edited, but by default, we're using a cutoff of 10%. And we're looking at the WHO drug resistance category for MDR-TB. So starting on the right, we're looking at a gene map of MTB. We can see the drug resistance genes, their names at the top. The green indicates that there is no drug resistance mutation found. Red indicates that there is a drug or loss of function mutation detected. And we can actually see then the position of that variant. The ones in gray indicate that it's low coverage over one or more drug resistance positions. So if we look at that red line, that's showing us the cutoff. And as you can see, for some of the gray ones, it dips just below that cutoff. So it's been called as gray as a low coverage region. And then finally, in the actual reporting, we can see the first line and second line MTB drugs, as well as the resistance or susceptibility call or low coverage call. And then again, showing the specific gene and mutation that was called in that position. So that comes to the end of the talk. I just want to cover this final slide. So if you're thinking of going and analyzing data or starting a sequencing project, it's really good to start thinking about your analysis before you even go ahead with sample collection and sequencing. So you should know things like, what data type do you have for analysis? Are you starting with raw data that needs to be demultiplexed? Do you already have FASTQ files and you just need an alignment tool? Or have you already done variant calling and you just want to be able to determine the clinical interpretation of those variants? It's good to know your starting material. For example, if we're doing DNA variant calling, you obviously want to start with DNA. If we're doing RNA for gene expression or RNA for fusion analyses, you need to start with RNA. Or you're doing liquid biopsy, where you would start with something like cell-free DNA or cell-free RNA. You want to know what platform was used. Short and long read give very different read lengths, which need to use different alignments and mapping tools. So you must understand the research question and why you're selecting long or short reads. You also should have some idea of the type of variants you want to detect, whether it's SNPs, indels, CFVs, certain biomarkers, or you want to perform other types of analyses like de novo assembly, gene expression analyses, or splice sites. All of these might use different tools that need to be optimized for your specific question. Another big thing to consider is your current bioinformatics expertise and your ability to interpret those results. You might need to reach out to people for assistance or use tools like SeqOne that can really assist you in interpreting what the variants mean. If you're running any of these pipelines locally, you need to understand your access to what computational hardware is available, things like Linux servers for running specific pipelines that require Linux, or high performance clusters or computing that's required for large data sets, things like single cell analyses, where we're dealing with huge amounts of data that can't typically run on your laptop or basic PC. And then finally, something that a lot of people don't consider is NGS has huge file sizes. So we're talking gigabases, terabases, and multiple different file types. So a FASTQ file is going to be much larger than a VCF file. So you might only want to store the VCF and delete the FASTQ. However, that means you can't rerun your alignment and variant calling because you've deleted the raw data or the FASTQ. So you need to consider, how you're going to transfer your data and where you're going to store your data, and most importantly, what data to actually keep. You might be using a sequencing service locally or overseas, and you need to get that data sent to you. So you need to consider download times, transfer times, and things like that. Thank you so much for listening. That brings me to the end of this talk. I'm happy to take any questions if you have.